Good morning and good afternoon. Once again, my name is Dr. Khomoso Makaula. I'm the Senior Science Manager with ASLM. I would like to welcome you to this very exciting session of our webinar series. Today, we are going to be talking about the ABCs of product development. At ASLM, our vision is to have a healthier Africa through access to quality laboratory services for all. I believe this is now a vision that you are very familiar with. Our goal is to advance the laboratory profession, practice, science, and network in Africa. And this aligns very well with our theory of change because as ASLM, we want to have a sufficient and professionalized workforce that is governed and regulated. We also want to have diagnostics that are available and scaled up where and when they are needed, affordable, and also that this should meet global health standards. Many of you might be very familiar with the African Union Agenda 2063, the Africa we want. In one of their goals, the second goal of that strategy is to have a well-educated citizens and skills revolution, which is underpinned by science, technology, and innovation. And under that goal, one of the theories is to have education and science, technology, and innovation-driven skills revolution. Under this strategy of Africa that we want, there is also goal number 4 that talks about transformed economies, where Africa, we want to drive our manufacturing and industrialization using science, technology, and innovation. So as ASLM, we are also contributing to this bigger vision of Africa. Hence, we have these webinars. Some of the expected outcomes is to have enhanced the knowledge and skilled laboratory professionals, to strengthen collaboration and networking within the laboratory medicine community. And I want to say to the upcoming scientists, we want you to dream beyond just the tests, the abnormal and normal tests, reports that you are issuing on a daily basis. But we want you to think about how you can change the diagnostics landscape in Africa. We also have our seasoned scientists who are experienced, and we also want to work alongside with you to mentor, to guide the next generation of scientists. And to the policymakers, we know that laboratory is more than just the numbers of samples that are run in a day. And we want to call on you to expand the horizon of what can be achieved through diagnostics in the healthcare system. Ladies and gentlemen, today I'm very excited to introduce to you one of our very learned, very experienced scientists in the African continent, Dr. Sheikh Shan. I believe I pronounced the name correctly. He has trained at the Santa National Research Scientific at the University of Toulouse in France, where he graduated with a PhD in 2013 in structural and functional biology. His PhD research was focused on the dynamic analysis and assembly of site-specific DNA recombination machineries using single molecular techniques. And from 2014 to 2016, he worked at the electronics department of the French Commission for Atomic Energy and Alternative Energies in Grenoble, France, as a postdoctoral researcher. There, his research efforts were focused on bio-inspired technologies for molecular electronics. In 2016, he joined the Institut Pasteur du Car, that we commonly call IPD, as a researcher in innovation and diagnostics. And his primary interests were centered on innovation for global health, particularly on the development and evaluation of point-of-care tools, digital health for biosample transportation, and banking and rapid extraction method for nucleic acid amplification. So since 2020, he's been leading the Diotropics Laboratory, a social venture established by IPD, the Muro Foundation, the Foundation for Innovative New Diagnostics, and IPD. And in this department, he focuses on development and manufacturing of diagnostic tests for neglected and epidemic diseases in Africa. So help me welcome Dr. Sheikh, as he's going to take us through his experience. You will realize from his presentation that there's already a lot that is being done in Africa. We need to support each other. We need to learn each other so that we can realize the vision of the Africa we want. Dr. Sheikh, over to you. Thank you.
Thank you very much, Dr. Commerso. Thank you to all the ASLM team members for this kind invitation. Today it is my pleasure to share some insights on the ABCs of diagnostics product development. My name is Sheikh Tijan Jain, and as said Dr. Commerso, I'm leading Diotropics, a manufacturing facility and R&D facility for diagnostics located in Senegal. So I'll share my screen. Today, I will divide my presentation in two parts. The first part will be focusing on sharing some insights about the Institut Pasteur de Dakar, its history, and how we went from laboratory works to product development. And then I will dive into the diagnostics product development phases. So IPD has a Center of Innovation. It was created in 1896 with the first laboratory in the north of Senegal in Saint-Louis. And then the laboratory was moved to Dakar in 1924. And in 1927, IPD discovered the yellow fever virus. 10 years later, it started the manufacturing of the yellow fever vaccine. And in 1966, the vaccine was granted, that was pre-qualified by WHO. And I think as we speak, it's the only human vaccine that is WHO pre-qualified in the African continent. In 2009, it transitioned from a from subsidiary of the Institut Pasteur de Paris to a foundation. We were run by the Senegalese law. In 2020, we launched the first diagnostic manufacturing, which is a second health product manufacturer at IPD. And in parallel, we also started the construction of what we call the vaccine pool to expand our vaccine manufacturing capabilities. And also IPD was granted the Galien Prize in the category of the best medical technology for its COVID-19 product. And since 2022, IPD has been investing in genomic algebra, in education centers, but also in high-tech laboratories for biological and RNA manufacturing, but also vaccine research. In a nutshell, IPD is 87 years of vaccine production. It has 12 reference laboratories, whether they are from WHO, FAO, Africa CDC, and ECOWAS. It also includes 50 sites for sentinel syndromic surveillance in West Africa. It has supported more than 40 countries in terms of epidemic management and outbreaks management. IPD also is very active in the research side with more than 100% research articles published in 2024 and more than 1.5 million COVID tests distributed during the COVID pandemic. So I would like you to focus a little bit on the public and the surveillance side, on the surveillance system that we have set up. It is a system that allows us to detect very early unusual health events, whether they are peaks of febrile disease, peaks of other syndromes. Here, I want to give the example of yellow fever. In the map here, we can see that in 2015, we had a hotspot of dengue, just one hotspot in one region, very close to Dakar. In 2016, it was the same dengue at same hotspot, but starting 2017, dengue has been detected in several regions in Senegal. So dengue is now endemic in the Senegalese country. What is interesting is if you look at the other map here, if you look at the other Senegalese map, we can see that dengue is not the only arbovirus circulating. Zika, yellow fever, chikungunya, and many other diseases sharing similar symptoms are circulating, which means in order to detect dengue, we need to do a differential diagnosis. And this sentinel system has also been expanded to neighboring countries in order to allow the same disease arbovirus surveillance. We are also very actively working on climate change in order to see how that change can really affect the circulation of this disease. So because we need this differential diagnostics, and we also were already manufacturing the yellow fever vaccine, we wanted to be able to detect the disease and also to better target our immunization campaigns using the vaccine. So that is why we have decided to work on the yellow fever RDT. And this is in line with the I strategy, which is the eliminate yellow fever epidemics by 2016 strategy set up by WHO, which means we know that there is a demand, there is a need of a yellow fever RDT. We know that there is a demand, but also in order to better assess the market for this product, we are not only limiting ourselves to Senegal. We are working with other stakeholders. I want also to share here the example of COVID-19. In the surveillance system, IPD is monitoring respiratory disease, particularly influenza. Here in purple, we can see the circulation of influenza A and B, and in red we can see the circulation of COVID. We are seeing here that they don't necessarily overlap, and since they share similar symptoms, here also the need for differential diagnostics is very critical, and that is why another reason why IPD has been investing a lot in working with its partners on a COVID-19 RDT. Another example, and this is the last example, so measles. The Senegal has faced several measles outbreaks despite several immunization campaigns. And we know that the measles vaccine is very efficient, yet we still are seeing cases in several regions in the country. So we have decided to work on develop a measles product that will allow us to detect the IgM, the antibody against measles. Here in this graph in red, we can see the number of measles cases shared by the Minister of Health. And we worked on a product. We generated a first prototype and deployed the prototype in the field for a pilot testing. And by just being in one side where there was a big hotspot of measles during 2 weeks, we have seen a peak in the number of measles cases. This not only demonstrates the usefulness of the product, but also it shows that the Minister of Health is underreporting the number of cases. And this is another reason for IPD to really invest in the development of this product. And besides Senegal, there's also another strategy, which is coming from the measles and rubella strategic framework. This is from Gavi and WHO. They are looking for an RDT to improve the precision, timeliness, and coverage of surveillance to support efficient and targeted immunization efforts. Instead of vaccinating everyone, it's easier when you don't have enough resources to deliver the vaccine to have a product that will help you really know exactly where your hotspots are in order to better streamline your resources, your vaccine. So we are also very much active in the field of product development because we believe that is the way forward to building resilience and sovereignty. IPD wants to be a first detector and a first responder by leveraging the 4Ds, which stands for detection, discovering, development, and delivery. We are detecting several pathogens across the 42 Senegal sites in 10 regional sites. We are processing daily data, processing weekly samples in the reference laboratories, and also deploying mobile laboratories to decentralize the diagnostics. IPD also discover new pathogens using its sequencing platform on human and animal samples, but also by running genomic and immunological assessment. And it's using all of this know-how to develop product using its bioprocessing and R&D laboratories for epidemics. It has a clinical trial team that really allows the validation of this product for regulatory approval. And currently, the two products, as I said earlier, that IPD is working on are diagnostics and mRNA vaccines. We also have a cell-based system. And once these products are developed, IPD will manufacture it using a very responsive manufacturing capabilities in order to be able to manufacture low volume and also high volume product. And this can only be achieved by investing in the human capital development for biomanufacturing, but also by manufacturing locally, because we believe that decentralizing production is very crucial to build to increase access, but also build trust with local communities. In order to locally manufacture the diagnostics, we created what we call Diagnostics, which with the sole purpose to promoting access to diagnostics. It has been possible through the collaboration of 4 visionary partners, which are the Institut Pasteur de Dakar, the Foundation for Innovative New Diagnostics, the Media Foundation, and the Institut de Recherche and Development. The positioning of Diagnostics is very clear. Its goal is not to compete with big manufacturers and generate a lot of profit. It's rather to have a public health impact by focusing on tests that are not available in the continent, or tests that are available but not satisfactory in terms of performance, or tests that are available but not affordable. And it does so by leveraging the lateral flow technologies, and also by leveraging industrial partnerships through tech transfer, but also building enough resilience in order to develop its own product. The business model set up by Diagnostics is quite unique. It's a business model where the demand is driven. It's a business model which is based on a demand-driven aspect, which means that Diagnostics is not manufacturing product and chasing clients, because it hasn't invested very much in the sales and the marketing team. So Diagnostics will manufacture a product when there is an express demand, and that demand can come from several parties, whether they are African Union member states, IVD contract manufacturers, NGOs, charities, etc. And in this business model, there will be members that will support the platform by paying a yearly subscription, which we call the membership fees, and that yearly subscription will cover the fixed cost of the facility, salaries, the cost of the facility, etc. And the countries for which a membership fee has been paid can access directly the Diagnostics portfolio by paying more or less the manufacturing fees. Let me give you an example. During the pandemic, Africa CDC and the West Africa Health Organization paid the membership fees for some countries to access the COVID-19 product. Because there were membership fees have been paid, those countries could access the COVID product by paying $2.5. The test price never exceeded $2.5. So this is a business model that we call a Netflix model, but there are many other business models that several companies apply. Currently, we have 2 facilities. One facility here that is dedicated to R&D and tech transfer. And during the pandemic, Fine and United invested in another facility with IPD for manufacturing. Here, we can manufacture up to 75 million RDT per year. And I'm showing you 2 examples of a product, the Say2 COVID-19 antigen product, which is already in the market, and another product, it's a yellow fever antigen RDT that currently we are working on with the support from Fine. Now, IVDs. I think I have shared enough information just to show how IPD is working on business modeling, market assessment, leveraging its surveillance systems. But now, what does it take to develop a product? What does it take to develop an in vitro diagnostics? So first, what is an in vitro diagnostic? These are test kits, reagents, control materials, calibrators, intended for use in vitro for the examination of specimens derived from the human body. The IVDs are part of the medical device, but it's a section of medical device because it doesn't go into the human body or interact with the human body. So all the rest of the medical device that are not in vitro diagnostic interact with the human body. So it's very important to make here the difference. The purpose of IVD is to provide information concerning a physiological or pathological state, a congenital abnormality, to monitor therapeutic measures, or to determine compatibility. I can give some example: the blood glucose monitor, pregnancy test, the COVID-19 antigen rapid test, but also extraction kits for PCR. This is an example from QIAGEN. IVD are very important in the healthcare system because they allow to diagnose disease. They are essential for early detection and accurate diagnosis of disease. The example I would like to share here is the differentiation between viral and bacterial infection in order to reduce antibiotic use. When you have a fever, when you have an infection, it's very critical to know exactly whether it's an infection by a virus or by a bacteria. If you don't have a tool in order to do the diagnosis, you may use antibiotic that will be of no use, and that can have an effect of antibiotic that can lead to antibiotic resistance. IVDs are also used for treatment monitoring. They guide patient management and therapy effectiveness. For example, HIV viral load. When someone has HIV and they get a treatment, there will be HIV viral load will be monitored just to make sure that the treatment is being efficient. And usually it's done using PCR or other techniques. IVD also plays a vital role. IVD also plays a vital role in disease surveillance and outbreak control. For example, during the Ebola outbreak in between 2014 and 2016, the first patient that had Ebola was people who thought that that person had malaria. Why? Because Ebola can share similar symptoms with other febrile illnesses, and having the right tool in order to do a very accurate diagnosis is very critical. IVD can also be used in personalized medicine in order to enable tailored treatment based on individual genetic and molecular profiles. For example, one may want to identify relevant biomarkers to deliver a very specific treatment to individuals. So what does it take to develop an IVD? In order to develop an IVD, we need to go through what we call the product development lifecycle management, or basically a design control through the design and control phases. There are several phases. The first phase, which is very obvious, is the planning. Then there is the concept and feasibility, which is the first phase. And then design and development, verification, validation, design transfer, and finally market entry. So I'll explain better what these phases, or the goal of these phases in the future slides. And these design controls will allow you to ensure that the product that will be developed and put in the market will be safe, reliable, and will meet the performance standards. They follow the ISO 13485 standard, or they can, when it comes to Europe or other countries, or the FDA 21 CRF 820, and this is for FDA. These are quality management systems, but for different regions, different settings, but they share the similar goal. The ISO 13485 standard ensures that medical devices meet both customer and regulatory demands for safety and efficacy. The 21 CFR Part 820, which soon will be called the Quality Management System Regulation, starting in February 2026, ensure that medical device companies establish a quality management system that enables the delivery of safe, effective, and compliant products. So here we can see that they share they have similar goals. And throughout your design control, you need a very strong risk assessment, because remember, your goal is really to develop a product that is safe, reliable, and perfect. The first phase, zero, very obvious phase, as I was saying earlier, is the planning phase. It includes defining the project goals and expectations. You also need to allocate your resources by ensuring the team, by setting up a well-capable team capable of delivering the project. And also you need to define very clearly their roles, the role of the team members, their responsibilities, and you also need to allocate budget. You also will need to make sure that the project will follow a defined timeline for delivery. And you also need to go through your risk assessment and mitigation plan in order to manage and mitigate delays, budget over, etc. So the risk should be assessed and mitigated throughout the entire product life cycle. For the rest of the phases, so I'll share the example of dengue diagnostic. Let's say we want to develop a dengue diagnostics to detect the acute infection in human. So we'll go first through the phase one, which is a concept and feasibility. First, we need to identify the need. We need to identify the unmet need by pinpointing the gaps in existing diagnostic solutions. Then we need to analyze the market by assessing market size, competitive landscape, and target user profiles. We also need to engage stakeholders to gather insights, and the stakeholders can be clinicians, patients, regulatory experts, or other stakeholders. In the phase one, we will also need to define a preliminary business case by evaluating the commercial viability and the strategic fit of the product that we want to develop. So in terms of need, if you look at for dengue, for example, here in the left, this is a picture showing the different symptoms that we have for dengue. This comes from the CDC website. We can see there are several symptoms like eye pains, muscle pains, bone pains, fever, headache, nausea, vomiting. But we know that these symptoms are not only seen in dengue infection. They can be seen in malaria infection. They can be seen in chikungunya infection. So you need a differential diagnosis. And also, dengue can be is very similar to other arboviruses such as Zika, etc., yellow fever. And because they are very close, differentiating them may be very tricky. And there is a gap here that we need to fill in terms of diagnosis. Now, to assess the market size, I'm giving you an example from Bateta, so published in Nature in 2013. Here, the authors estimated the burden of dengue in 2010 per continent. And we can see that the apparent burden for dengue case is 15.7 million cases, and the inapparent dengue cases is 40.4 million cases. So the author did that by leveraging several resources. First, the clinical trials on dengue that were being run worldwide. They looked at disease prevalence, disease incidence. They look at the number of cases that have been shared by Ministry of Health, and they use some modeling. So there are many resources and insights that people can use in order to assess market size. Now for the strategic fit and commercial viability, here I've shared some pictures of dengue surveillance in Senegal. So we saw here that Senegal is already doing dengue diagnostics, but also differential dengue diagnostics, but using here PCR, which means if you want to develop an RDT, we know that there's already a market because Senegal is already paying for this. And the strategic fit for IPD is very obvious because it's critical to have a dengue, a very good and portable dengue product that can reach low resource settings because IPD has a public health mission. which means we have the public admissions and it's a strategic fit. We also know the product can be commercially viable in the Senegalese market. If you want to look at a market outside Senegal, we need to look at the same kind of data in other neighboring countries. Still in phase one, we need to assess the technology by doing a technology scouting, by exploring innovative scientific principles and platforms. And then we need proof of concept studies by running initial laboratory experiments to experimentations to demonstrate technical feasibility. Here we select the analytes, the biomarkers, the reagents, etc. We also need to go through a risk assessment in order to identify potential technical, scientific, and operational challenges and mitigation plans. And the risk assessment can be done by using the failure mode and effect analysis that we call the FMEA, or the ISO 14971 standard, which is the standard dedicated to risk assessment. Still in phase one, we need to assess the patent landscape and potential for novel IP to protect, in order to protect the product that we are developing, but also to avoid patent infringement in case someone else had already the same idea and protected that idea. So now for proof of concept, we want to develop a product that will detect acute cases of dengue. We know that the NS1 biomarker is a good biomarker for acute dengue cases. So here, what we need to do is just to have a buffer solution that will be spiked with the NS1 antigen. And then we need just a strip that with a test line that will be loaded with antibodies against the NS1. Ideally, we want to do this test using a plastic housing. The test will be in a plastic housing. We want to use not buffer solutions, but blood samples, serum or plasma samples. But at this stage for feasibility, it's not needed. We just need to make sure that we can load the sample onto the strip and we can detect the biomarker onto the strip. So this is your proof of concept. And once you can achieve this kind of thing, you are good to go ahead in your development. Now for risk assessment, here I would like to talk a little bit about the FMEA, which stands for failure mode and effects analysis. So this is a concept that will allow you to detect all the failures, potential failures that your product can face. And also you will need to understand the effect, the impact on the end users. In order to do that, so there's what we call the risk priority number, the RPN. The higher the RPN, the higher the risk is. And if it's very high, so that means that the manufacturer needs to pay attention and it needs to deal with this risk accordingly. The RPN is calculated by multiplying the severity by the occurrence by the detection. The severity is the seriousness of the failure and its consequence on the end user. And we have the occurrence, which is the likelihood of that failure to happen, and the detection, which is the likelihood to detect that failure. So FMEA is really a tool that will help you assess all failures and mitigate them, but they are very specific to product designs. However, a product may not have any failure, but still present a risk, and that risk will not be handled by FMEA. That is why you need a risk management plan through the ISO 14971. So this standard, which is very broad and more complete than FMEA, will allow you to perform your risk analysis to evaluate the risk and control them. Once the risks are controlled, there will still what we call a residual risk. And here you will see, the manufacturer will check whether the residual risk is acceptable or not. If it's acceptable, it's okay. If it's not acceptable, it needs to be handled. And also here we'll go through a risk management review to make sure that all the risks are assessed, they are detected, and they are controlled. And after doing the design, and after the product is launched, this is what we call the production and post-production activities, which mean the risk assessment will still continue, because we want to make sure that once your product in the market is still safe, reliable, and performant. For example, during the pandemic, the COVID-19 product in the beginning was working very well, and then we have variants, which mean the product will lose performance. And by going through your risk assessment, you can detect those loss of performance, and that will help you fine-tune your product. Now we go to phase 2, which is the design and development phase. Here, we start with the design inputs. Your design inputs clearly define the purpose, target population, and testing environment. It documents what the users need the device to do, and it will convert the user needs into measurable requirements. We include regulatory, safety, and usability criteria. Here also we'll choose the detection methods, whether they are colorimetric, fluorescence-based, or PCR-based. We also define acceptable range for key metrics, such as limit of detection, runtime, performance, and all the design inputs will be documented in a design input requirements file. Because your design input will generate some other documents that we call design outputs, they will be put together in what we call the design traceability matrix. And then still in phase 2, you will now develop your essay. You have your input, you develop your essay. You develop it by selecting your components, by sourcing and evaluating raw materials, reagents, components. And also you will go through an iterative prototyping by creating multiple versions of the device, learning from each iteration in order to optimize sensitivity, specificity, and stability. Then you have your design outputs. You have inputs and outputs. Your design outputs will be the documents that will show that the design inputs have been implemented. They are product specifications, for example, the raw materials or the bill of materials, your components, sub-assemblies, packaging, and labeling specifications. They will include your instructions, the design drawings, schematics, and manufacturing instructions, and also all the reports and validation documentation of your risk management plan. Now, some example of user needs, design inputs, and design outputs. First, user need, probably this is just an example. Your dengue test must detect acute dengue cases with high accuracy to ensure clinical reliability. Your design inputs can be the sensitivity and specificity will be superior to or equal to 98% in whole blood and capillary samples. And your design outputs will be validated antibody pair targeting your biomarker NS1, integrated into strip with optimized capture line. And here you will verify it by performing internal quality control and running a clinical trial. Second user need example of a user need, the sample collection must be minimally invasive and safe for patients. The device as an input shall accept 20 to 50 microliter of capillary blood from a finger stick with no exposure risk to user. And your design outputs will be a preloaded Lancet and integrated buffer sample port system in sealed unit, and you will validate it for user safety and biosafety compliance. Final example, your device must function reliably in low resource settings with limited electricity. Inputs, the device must operate without mains power and store stably between 2 and 40 degrees Celsius for 12 months. Your output, a battery-operated device using lyophilized reagents with desiccant packaging, validated for stability at high temperature and humidity. Then we have the phase 3, which stands for design, which are the design and verification. We start with the verification, which is the confirmation by objective evidence that specified requirements have been fulfilled. Here you will use test methods. You will generate pilot loss manufacturing to ensure that your manufacturing process is robust. You will perform functional testing, you will run your assess your performance. You will conduct reproducibility and repeatability studies, stability studies, stress testing, and you will detail all your test protocol results and deviation reports. You will assemble that documentation that will go into your verification documents. And again, you will need to go through the design traceability matrix, because remember, here you are ensuring that every design output can be traced back to its corresponding input. And when putting together design input and design outputs, you need the design traceability matrix. So this document is still needed in the design verification phase. Now you have a product that has been developed, it's verified, now it needs to be validated. The validation is a confirmation by objective evidence that the requirements for a specific intended use or application have been fulfilled. In order to do the validation, you will need a study design by developing rigorous protocols. You will need ethical approvals from your IRBs or ethics committees. You will need to recruit patients, collect data and analyze them. You will need your clinical performance report, and you will need your user acceptance testing, which will involve end users to confirm that the device meets their needs. Now the design transfer. We have a product, it's developed, it's verified, it's validated. So now we are ready to move to go to full manufacturing. And this is a step that is called design transfer. This is a here you will need to validate your process by ensuring that the manufacturing process consistently produces a product meeting specifications, and you will generate your design master record. The design master record is your recipe. It's the final documentation handover that will include your design drawings, your QC methods, manufacturing instructions, packaging, and labeling. Once you have the design master record, it means that you can use it, you can share it with stakeholders, with other colleagues, and they will use that to manufacture the product. But these are just documents. How do you ensure that people will use the documents very efficiently? You need to do that by adhering strictly to quality control during production. You need to implement a very strong quality control process. They can be in-process control or control of your final product. And at this stage, any design changes must be controlled through a formal change control process. Because your design is frozen, you have a product that now goes through full manufacturing. All change need to be handled by the change control process. Phase 5, which is the market access. Here you need, if you want, the first step in accessing the market is understanding regulations. So there are several regulatory bodies. For example, in Africa, we have SAHPRA. In South Africa, we have FDA in Ghana, the Poison Board in Kenya, ARP in Senegal, we have FDA in the US, CE mark. There are many others. And each regulatory agencies will classify your IVD based on the risk classification. For example, FDA has 3 classes, class 1, 2, and 3. The higher the class, the higher the risk. In EU, European Union, we have the class A, B, C, and D. Here also, the higher the class, the higher the risk. And here also, you need to adhere strictly to the standards like the ISO 13485 or your quality management regulation system when it comes to FDA, or maybe other quality management systems depending on the regulatory bodies that you are targeting. Just a couple of examples here of IVDR, which is the in vitro diagnostic regulation. That is the regulation in Europe for in vitro diagnostics. You can see that there are several classes, and depending on your intended use, your test will fall in one of these categories. For example, our dengue product will fall under the class C here. And if you are in a class C in Europe, you need to go through a notified body approval in order to be able to release your test in the market, which means a notified body need to assess your dossier and make sure that the test is safe, reliable, and performant. For example, in another hand, when you have a class A product, which are specimen receptacles, prepared selective culture media, it is low risk, so you can just use a self-assessment. You don't need a notified body. And if you go to FDA, you have the lower risk, class 1, 2, and 3, as I was explaining earlier. And for class 1, which has usually toothbrushes, etc. So you don't need any FDA approval. For class 2, you need an FDA clearance, which means that you will need to go through the 510K, which is a process designed by FDA for you to show that the product you are releasing in the US market is very similar to a product that has already been approved by FDA. If you can demonstrate that, you just need a clearance. But for high risk product like the class 3, you need to go through an FDA approval, which means you need to go through the clinical trials, etc. So your lead time will be longer for your dossier to be accepted, and also it will cost you more money. In Africa, we have 54 countries, 54 regulatory agencies, and they are divided in 5 regional regulatory agencies. So five blocks. So you need to understand exactly which market you're targeting in order to know which regulatory agencies you will need to go. And currently, there is through the African Medicine Regulatory Harmonization, the African Medicine Agency is working on a harmonization of the regulatory pathways in Africa, which means you may have a product registered approved in ECOWAS, and the dossier that has been put here, submitted to ECOWAS, may be transferred, for example, to the Eastern African Community Regional Economic Bloc. So once this is done, so it will fast track your dossier approval, and will cost less money, and the lead time also will be reduced. So again, still in market access, you need to prepare a dossier that will be submitted. There is what we call a STEP document, which is a summary of your technical documentation. So this has been put together by the Global Harmonization Task Force, which means the regulators have met and they have decided that your dossier, the dossier that you need to submit to your regulatory bodies, can be harmonized. And this is the STEP document is used in many countries around the world. Basically, it will the technical documentation that the manufacturers have generated and developed under a quality management system will go through the step. For example, here let me just take one example. The labeling that have been developed by the manufacturers will go through the set of labels and instructions for use in the STEP document, which means you need to check all the boxes here and tick the boxes and make sure that all of these requirements are available from the manufacturer side. But also there is something I would like to share. When dealing with regulation, you need pre-submission meetings, which means that you need to engage early on the regulatory authorities for guidance, because each regulatory agency has its own specifications and requirements, and you don't want to find out those after your product has already been developed. Because, for example, if your regulatory agency is asking for a stability degree of, let's say, 45 degrees Celsius, and your product has been developed to, to be in an environment not exceeding 40 degrees Celsius, that means you need to go back to your product development. It is something you would like to avoid. So understanding early on the requirements is really key to fast tracking your development and avoiding any mistakes later. Then the market access, still in the market access, you have the launch process. Here you will go through your marketing and sales strategy by developing your go-to-market plans, your pricing, distribution channels, customers, finding your customer, etc. You will need training and support. You will need to provide comprehensive training for your sales team and end users. You need customer feedback by establishing channels for post-sales support and feedback. You also obviously need a launch event to officially introduce the product to the market. In terms of go-to-market planning, there are several elements that the manufacturer could leverage, or some questions that the manufacturer should need to answer in order to define a very effective go-to-market strategy. The first one is related to the most attractive and viable customer opportunities. Here, the manufacturer will ask itself, what are the key customer segments for its portfolio and how significant are each segment? For example, if I am developing a product for global health institutions or global health buyers, I need to understand whether that product is strictly can also go to private market, or will that go to UNICEF supply division, etc. So really understanding the segment is important. And once you identify that, you need to define the right channels for distribution. You need to understand what are the optimal channels for each customer segment, including key stakeholders to engage to enable effective and efficient engagement and coverage. The third one is the right offering to maximize access. The question here that the manufacturer asks itself is how are the players in the market pricing their products, and how should it position its offering to maximize access to customers? The fourth point is a compelling value proposition. The questions are, what are the key purchasing criteria for priority buying channels across the key customer segments, and how best the company could position itself to win? Finally, the fit for purpose capabilities and organization need to be enabled. And here's a question that the manufacturer will ask itself is, what best-in-class capabilities will it need to effectively serve its key customer segments? And what are the must-have functions and roles, and how should the company organize them? Then once your product is launched, so you need to do your post-market surveillance. You need to have vigilance reporting by monitoring and reporting adverse events and product malfunctions. You need to handle complaint by systematically managing and resolving customer complaints. You will need a post-market clinical follow-up to conduct studies to gather real-world performance data, and you will need a continuous improvement by incorporating feedback into your product updates and next generation designs. Here you will use what we call CAPA, which is a corrective action plan that will be used to identify, analyze, and resolve the root causes of non-conformities, complaints, or adverse events related to a product process. And this is my final slide. Just wanted to highlight that the design control related process, whether it's based on the ISO 13485 standards or the FDA standards, follow from strict guidelines, and the different chapters and annexes can be, for the different sections, can be really found in this table. So this is the end of my presentation. Thank you very much. So I have added my email addresses here in case people would like to contact me later. And in the meantime, I'd be happy to respond to any questions. Thank you very much.
Yes, thank you very much, Dr. Sheikh, for such a very informative session. Unfortunately, we have very short time for Q&A, but we would also like for you, the participants, to help us in the evaluation of the session. Dr. Sheikh, I think one of the questions that I just remembered from the chat is whether there are any current openings for fellowships with IPD in manufacturing. And then the other question was around, I don't know if you can see the questions. The one around how do you use LFAs for routine testing? I'm trying to look for the question. I can't find it now. Yes, please. We have under 5 minutes left. If you can go through those questions that have not yet been answered.
Great. So in terms of fellowships, yes. So currently there is a fellowship with Stellenbosch University in South Africa. There are several of those. There is one that is targeting young professionals who would like to be trained in business development, product development. There's also an MBA, as I think the MBA has just been released this week. for young professionals. So I can share the link later to you, Dr. Komatso, and you can also share it to everyone. But it's also publicly available on LinkedIn. So I really encourage you to go and check, because I think these are very good opportunities.
Okay, thank you very much. So we'll include the link when we share the presentation. The other question is about, it says it has been said that one of the key opportunities to improve access to IVDs is the use of LFAs. However, LFAs can support rapid and decentralized testing, particularly during disease outbreaks. However, in the context of routine primary health care, LFAs may not be preferred option due to concerns around quality and reliability of testing. And given this, what is your perspective on the appropriate role of lateral flow assays in strengthening diagnostic capacity at the primary health care?
Yeah, I think that is a very good question. So is that, we know that lateral flow can have a performance that is lower than the performance of PCR, for example. But it's not always true. Currently, the measles IgM product that we are working on outperforms the ELISA IgM that is currently in the market. So obviously, this needs to go through an independent evaluation. But in some cases, we can have very, very, very good RDTs. But also, we need to understand is, even if PCR can give, for example, if PCR can give very, very, very good performance, you perform it once, let's say it will cost you $10. I'm just giving an example. If you have an RDT that costs you $1, but you can repeat it 8, 7, 5 times, you may outperform at different time points, you might outperform the PCR. And but yeah, so I think it's also due to how the RDT is seen, because I have seen myself many healthcare workers that will say, yes, we want the RDT, but we know that it has limited performance. How could we handle that, etc. So it's very cheap, portable, and currently we are trying to integrate molecular capability into lateral flow in order to have something even more performance, cheap, and handheld. So I have seen very good prototype. We have PhD students working on that. I have also seen several companies working on that. And the product I have seen, particularly in malaria, are very promising. You can integrate your molecular isothermal assays into your rapid test. And I think this may give you a stronger performance, and it also may increase trust in the product for healthcare workers.
Okay. Thank you very much, Dr. Sheikh, for such an informative session, and thank you very much, our participants. This is very encouraging, inspiring. So we'll be sharing more resources as we share today's presentation in terms of, I've seen also in the IPD side, they have training courses if you want to know more about product development. So we really do this to raise awareness, to inspire you, to venture into these new areas where we will be able to bring innovation in our African continent. So coming to our next webinar, we do have a webinar that's coming up on the 28th of July. We are going to be looking at molecular and precision medicine. These are very exciting topics. Some of us, maybe by the time we were in school, these were not yet covered. So it's time to learn and also to see how we can bring new innovations into medical laboratory. So thank you very much. The questions that have not yet been answered, we are going to post their answers, and share the materials together with presentations from today. So thank you very much for attending today's session. Bye-bye.